Hello everyone, my name is Beatriz Ribeiro and I'm here to present you my work called the P2X7 receptor modulates inflammation and collagen deposition in experimental chronic colitis and colitis with human IBD colonic myofibroblasts. This work has been developed by me and my group at the Federal University of Rio de Janeiro, Brazil. I thank very much the opportunity of presenting this work in the Purines 2021. So we are talking about inflammatory bowel disease, a chronic relapsing condition that affects the gastrointestinal tract. It encompasses mainly two diseases, Crohn's disease and ulcerative colitis. The pathogenesis of these diseases is not yet fully understood, but we do know that environmental factors, such as food antigens, microbiome, and immune modulators, create a pro-inflammatory response that self-sustains over time and can lead to fibrosis. The P2X7 receptor is a cell membrane receptor activated by ATP in the extracellular milieu. It acts as a damage-associated molecular pattern, and it opens a pore in the membrane when activated. It also leads the downstream pathways that cause the activation of the inflammasome made up by NALP3 and caspase-1 proteins. When activated, the inflammasome can cleave pro-IL-1 beta in its active form. Our group has been studying the role of the P2X7 in IBD for a while. The aim of this study is to understand the role of the P2X7 receptor in the development of intestinal fibrosis associated with chronic inflammatory environments, such as an IBD. For this, we induced chronic colitis in C57 black 6 male mice. We used dextrin sulfate sodium in the drinking water. The animals were exposed to a week-long DSS exposure, followed by 2 weeks of pause. This cycle was repeated 3 times, and the animals were previously dewormed. First, I would like to show you the survival rates and the body weight change. Notice a trend of decrease in the survival rates of the animals subjected to DSS, and we also did not see an increase in the body weight over time, as we see in the control mice. Further, when we look into colonoscopy exams, we see that the wild-type mice subjected to DSS present a higher colonoscopy score when compared to the other 3 groups. In this case, we can see bleeding, ulceration, change in the pattern of the mucosa, and inflammatory infiltrates that we not see in the knockout mice subjected to DSS. Next, we assessed the column stiffness by EUBM, that is, endoluminal ultrasound. In the middle of the image, we see the ultrasound probe and the layers of the mucosa and colonic wall. What we did was that we injected saline solution intrarectally over time, and we assessed the increase in the cross-sectional area. Notice that the wild-type mice subjected to DSS present a small variation in the cross-sectional area as we inserted saline solution, while in the other groups we see a higher variation in the cross-sectional area. Here we can see images from colonoscopy, EUBM, and histology. Notice that these animals, the wild-type animals, present alterations in the 3 images. In histology, we see a large colonic wall and a pink fibrotic tissue around the mucosa. We do not see such alterations as marked as in the wild-type mice when we look into the knockout mice. Here, we assess the histological score, where we can see inflammatory cells and ulceration in the mucosa. We also assessed the glycosaminoglycan density in the tissue, and we see that they are increased in the wild-type mice compared to the other 3 groups. This is a picrocyrus staining for collagen. Look, at a large staining for collagen in the colonic wall of the mice subjected to DSS when wild type. In the knockout mice, we do not see such an intense collagen deposition as we see in the wild type mice. So here are the graphs showing these results. We also assessed the apoptotic rate by tunnel assay, and we found similar results with an increase in apoptosis in the wild-type mice when compared to control and when compared to knockout mice subjected to DSS. We also stained NALP3 caspase-1 using immune histochemistry, and we found an increase in these stainings in the wild-type mice when compared to control and when compared to knockout mice subjected to DSS. The same results were found for stainings for NF-kappa B and ERK. Next, we assess cytokines by ELISA. Notice that TGF-beta and IL-10, both anti-inflammatory cytokines, are elevated in the wild-type mice when compared to control and when compared to knockout mice subjected to DSS. When we look into pro-inflammatory cytokines such as IL-1 beta, also interferon alpha, we found similar results. TNF-alpha, IL-6, and IL-17 did not present statistically different results. We also assessed PCR gene expression for similar targets such as NF-kappa-B, MAP kinase, TGF-beta, and metalloproteinases, that is, proteinases responsible for the turnover of the extracellular matrix, and we found similar results. At last, we collected biopsies from patients with Crohn's disease, ulcerative colitis, and control patients. We did immune fluorescence to stain P2X7 and to stain vimentin, a marker of mesenchymal cells. We do not see a difference in cells stained for P2X7 and vimentin at the same time in the three groups. But when we look further into staining of alpha SMA, a marker of myofibroblasts, we see an increase of cells positive for both markers at the same time in Crohn's disease and in ulcerative colitis when compared to control patients, and more so in Crohn's disease. As a result, we see that the knockout mice subjected to chronic colitis displayed attenuated clinical manifestations, as well as micro and macroscopic inflammatory alterations, colonic stiffness, and collagen deposition. Pro-inflammatory pathways were found decreased in knockout mice compared to control mice. Similarly, anti-inflammatory cytokines such as IL-10 and TGF-beta were also decreased in knockout mice. At last, human colonic myofibroblasts are overexpressed and colocalizes with P2X7 receptor in Crohn's disease and ulcerative colitis patients. I would like to thank very much my professors and the lab members, and also you for watching this presentation. Thanks.